So good morning, everybody. Monday morning, and I am Jana Hexter with the Northeastern IPM Center. And it is my pleasure to introduce you to the Northeastern IPM Center Research Update Conference. It's fun. I always enjoy doing these. We have another one coming up on Thursday, at the same time on Thursday. And before we get going, there's just a few quick things. If I could get there we go. We are recording this. It will be available next week at that link, and I'll also send you an email with the recording. We welcome your questions, and if at all possible, I ask you to put them in the Q&A feature, which if you scroll over and get that black box that comes up from Zoom in the middle, there's something that says Q&A. If you put your questions in there, it's easier for me to monitor them. We do have 5 minutes each half hour for asking questions. And also you can put things in the chat if they're as the videos are going along too. We'll do our best to monitor. I will leave that till later. I will stop sharing, and there's going to be a little Zoom wizardry going on as we switch between presenters. So our first presentation video is Jaime Pinero talking on multi-cultivar grafting, a novel low-cost grower-friendly attract and kill approach to manage key apple pest and it's funded through ARDP, and he is with the University of Massachusetts Amherst.
Good afternoon. My name is Jaime Pinero, and in 5 minutes, I will discuss a research project, which the goal is to develop trap cropping systems for multiple pests in apple. This research is in the context of eco-stacking. Basically, how can we maximize ecosystem services by increasing biodiversity in orchards, which is difficult to do. And we seek to integrate biodiversity in the form of genetic diversity within the rest of the cropping system. 5 years ago, when I came back to UMass, soon after that, I was meeting with growers discussing how can we make apple IPM more sustainable? How can we reduce inputs, for example? We have very powerful attractants for some insects, for example, plum curculio, but they're expensive. So growers are not using those lures for monitoring or for control. So can we develop a trap cropping system that is permanent, that it doesn't involve any lures, that is inexpensive, that is grower friendly, and work for the entire season? So this project is in collaboration with Jeremy Delisle and Heather Bryant from University of New Hampshire. And the idea was to graft individual trees in the perimeter of the orchards with six different cultivars. And those are reported to be attractive to plum curculio and apple maggot fly, but we're also including other insect species in the system. So I hope you will agree with me that grafting, the concept, is simple, inexpensive, and grower friendly. In 5 years, we have grafted more than 150 trees in more than 20 orchards in 3 New England states. This is a long-term project, and we're going to emphasize monitoring. Is this approach effective? We have done research for 3 years. In 2021, for example, you can see across all orchards in Massachusetts, when I combine all the injury in red astrakhan, injury by plum curculio recorded at harvest, 34% of the fruit has injury by plum curculio. Wixon, 31% injury. Stock fruit is the Macintosh, the Empire, the Cortland, which are original in the original canopy. So there is a lot of injury, congregation of injury in those canopies. This map shows an example. One block in Belchertown, where we have in blue trees grafted with multiple cultivars, some distance and 30 meters among them. You have in orange perimeter row trees, which are just in normal trees, non-grafted with multiple cultivars. And very quick, because I will not have much time, just every year you can see an increase in injury, an aggregation of injury in the grafted branches. Sometimes you see the same pattern in the stock branches. But in comparison, the perimeter row trees, which are in brown, orange, non-grafted, there is less injury. So it seems to be working. When I can show you now injury according to cultivar. So every year, you can see the average injury is higher in general in Relastracan Wixon compared to Davinet, for example, but also yellow transparent seems to be working well every year. Apple maggot fly, we are recording captures every week in unbaited red sticky spheres deployed in grafted versus non-grafted trees. Every week, we are counting insects with apple maggot fly on those spheres. And every year, you can see in the charts, we can see at least twice as many more flies captured on sticky spheres deployed on grafted versus non-grafted trees. So seems to be working. Wixon is a very interesting cultivar. I will be happy to tell you a story, but next time. But we're finding that Rosia pole aphid seems to be able to prefer some cultivars over others. So here is 2 weeks of research collected in 3 blocks. You can see that in general terms, there seems to be more injury numerically, sometimes significant, in ginger gold and red astrakhan. We need to do more research. We're also recording the parasitism levels of Rosia pole aphid in these different cultivars. Red astrakhan and ginger gold, numerically, there seems to be more parasitoids. We are also recording beneficial arthropods in those different cultivars. Here I am combining all the insects that we found, or arthropods that we found. So in conclusion, I believe that after 3 years of research, indeed, multi-cultivar grafted trees are attracting at least plum curculio and apple maggot fly in greater numbers compared to non-grafted trees. And this can allow growers to be able to monitor multiple pests across the growing season using grafted trees. So I would like to thank growers in 3 different states in New England, collaborators, students, and also the funding agencies.
Great, thank you very much. That was really interesting. So our next presentation is by Long He from Pennsylvania State University, University Park. And his presentation is Precision Spraying Technologies for Tree Fruit Orchards, and it was funded by ARDP.
Hello, good morning, everyone. So thank you for coming for the seminar. First, I want to thank you, the Northeastern IPM Center, for providing this opportunity for us to share our research updates. My name is Long He. I am an assistant professor in the Department of Agriculture and Biological Engineering at Penn State, and also a physical located at Food Research and Extension Center in Biglerville, PA. So before I go to the research updates, I want to just share some of our thoughts on the concept of precision spraying systems. You can see from this picture, a precision spraying system mainly including 2 parts. One is the detection and control system, and the other part is the spraying system parts. For the detection, we're normally using sensors like LiDAR sensor, camera sensor, or some other type of sensors to detect object in an agricultural environment. And variable rate control, that means we can control individual nozzles for the flow rates from those nozzles to fulfill the needs from this precision spraying. And targeted spray, so it depends, like we can target the whole canopy. We can also only target infection area within the canopy, or we can target flowers, fruits, and other objects if we are interested on those objects. And further step, we can further develop this spray system to more in-automation robotics system. So we can make this sprayer drive by themselves and without human operators on board. And now I'm going to talk about 3 different projects briefly. So the first one is an intelligent sprayer for orchard pest management. You can see from the left, this is a sprayer that we integrated a conventional air plant sprayer with a intelligent sprayer kits. So it is including a LiDAR sensor to detect the canopy density. So based on the canopy density, we control the nozzles and spray accordingly. We have been testing for a couple of years, and then we found that like we can save chemicals around 50%. And it depends on the tree structure and also the time of the season. The canopy density can change throughout the season. So it varies, sometimes save more, sometimes save less. But it definitely has a big chemical saving. And And also we add another system called inlet airflow control to minimize the drift. You can see this is an Irish damper that we installed in the back of the sprayer that can actually adjust the opening of this Irish damper based on the tree canopy density. And based on the test, we found like the drift can be reduced dramatically if we consider the tree density during the spray. And then the second one is a robotic sprayer system for tree fruit orchards. You can see from the left side, this is how the sprayer looks like. And we did test in an apple orchard, also in a vineyard. And you can see from this drone footage, you can see how the sprayer operates in the field. So it using the ArcticGate GPS for the presetting path and in orchard map. So you have presetting maps, so the sprayer can driving along the fields with those presetting maps, and also can adjust canopy area coverage. So the nozzle range can be adjusted to cover the canopy that for the particular field. And it also has the potential for the spot spraying if we add some detection sensors, like I just mentioned, like LiDAR or camera sensors for more precise control. And last one is a precision spray system for target spraying applications. So for now, we only test this system for blossom thinning. You can see this is the sprayer that's using an unmanned ground vehicle and has a camera system here and a nozzle here. So the camera actually can detect the flower clusters, so we know the location of those flower clusters, and then we can spray with this nozzle very precisely. And like I said, this right now we only test in the blossom thinning, but also this can be potentially used for like precision weed management in orchards, so we can detect the weeds in the field and then spray more precisely with those weeds. And it also can be used for target spray for like precision disease and pest control that we can target only for those infection areas that we detect from those camera sensors. I think that's all for my research updates. Thank you so much for listening, and I'm happy to take any questions you have. Thank you very much.
Great, wonderful, thank you. And feel free to put your questions in using the Q&A feature, or if that is not working, into the, and I'll try and pay attention now. So our next presentation is from Kurt Vollmer, using spring-seeded grass cover crops to reduce herbicide inputs in plasticulture production. And Kurt is with the University of Maryland, and this research was funded by ARDP.
Weed control in the row middles of plasticulture production systems can be challenging given that fewer effective herbicides are available for control. This problem becomes worse when herbicide-resistant weeds such as Palmer amaranth are present, which further limits available control options. Previous research funded by the Northeast IPM Center showed that spring-seeded grass cover crops can be effective for early season weed suppression, but they do not provide full season weed control. Therefore, we hypothesized that both the cover crop and any weeds present could be managed with a single application of an effective post-emergence herbicide. Following this application, any remaining cover crop residue would continue to aid in weed suppression. Plastic was laid and cover crops were seeded by hand approximately 4 weeks prior to planting each cash crop. Cover crop treatments consisted of cereal rye, spring oats, or a combination of cereal rye plus spring oats. A no-cover treatment was also included. Cash crops were planted prior to cover crop termination. Watermelon was the selected cash crop at our Maryland site, and cucumber was the selected cash crop at our New Jersey site. Cover crops continued to grow for another 3 to 4 weeks before being terminated. First, cover crops were terminated by simulated roller crimping with the tractor. Afterwards, either paraquat or clethodim was applied using a shielded sprayer. A no herbicide treatment consisting of rolling only was also included. In order to determine if additional residual herbicides were needed, a treatment of fomeciphen plus S-metolachlor or a no residual treatment was applied by hand 24 hours after termination. Moving on to our results, cover crops reduced overall weed density prior to termination in both studies. In the cucumber study, there were no differences in weed reduction due to individual species, but in the watermelon study, the oats plus rye treatment had fewer weeds compared to the rye-only treatment. In the watermelon study, the main effects of cover crop, termination method, and residual herbicide were significant for late-season weed density, but there were no interactions. The presence of a cover crop reduced weed density compared to no cover crop. A greater reduction in weed density occurred when the cover was terminated with paraquat compared to clethodim or by rolling, and a greater reduction in weed density was observed when a residual herbicide was included. Similar results were seen with overall late-season weed density in the cucumber study. Cover crop treatments had fewer plants compared to no cover crop treatments, and terminating the cover crops with paraquat resulted in fewer weeds compared to terminating with clethodim or by rolling. In the watermelon study, there was a significant cover crop by termination method interaction for late-season weed control. While control was greater with most cover crop termination combinations compared to no cover crop treatments, terminating with paraquat was more consistent across cover crop species. And when a cover crop was terminated with paraquat, control was greater compared to applying paraquat to a no cover treatment. Results were observed for late-season weed control in the cucumber study as well, showing that cover crops terminated with paraquat had significantly higher weed control compared to terminating a no-cover treatment. Only the effect of cover crop was significant for crop yield in both the cucumber and watermelon studies. In the cucumber study, yields were greater in the cover crop treatments regardless of species compared to the no-cover treatments. In the watermelon study, oats and rye treatments had greater yields compared to the oats plus rye and no cover treatments. These studies support previous work showing spring-seeded grass cover crops can successfully be used for weed control in plasticulture production. This work also demonstrates how effective termination tactics can be used to both manage cover crops and weeds, allowing for extended control later in the growing season.
Great, thank you very much. All right, we have one more presentation, and then we'll have a little bit of time for a Q&A. And the next presentation is by Leo Kerner, who is with the University of Maryland. And he's going to be talking about developing a perennial living mulch system to manage insect pests in northeastern cantaloupe fields.
Hey everybody, my name is Leo Kerner. I'm a graduate student in the Department of Entomology in Dr. Sarita Hooks' lab at the University of Maryland. And today I'll be talking about, I'll be giving a research update for my master's thesis project, which considers red clover as a living mulch and its effects on weed suppression. And just to mention, this project was funded by the USDA's National Institute of Food and Agriculture and the University of Maryland's Department of Sustainability. So what is living mulch? So a living mulch is a secondary crop that grows between the cash crop rows. And its goal is to provide some ecosystem services. This could be increasing the abundance of natural enemies. It could be reducing the accessibility that insects have to your crops, or it could be to reduce weed suppression or to increase weed suppression, I guess. So we selected red clover this previous summer for a variety of reasons, because it is fast growing, it's pollinator friendly, it's nitrogen fixing, it's not supposed to be light competitive because of its relatively short stature, and it's understudied. So this was our setup for this previous summer. This was a complete split block design, which was replicated 4 times. And we had 2 whole plot treatments. So our first whole plot treatment up at the top is our control. It was a bare ground plasticulture system. So we had black plastic over the crop rows. Our cash crop was cantaloupe. And then in our second whole plot treatment, we did red clover, which was drilled at 12 pounds per acre at 6-inch drill spacing. Additionally, we were interested in comparing chemical and cultural techniques. So we had each of our whole plots split into high and low chemical input subplots. So our high input subplots received a pre and a post plant herbicide application, and our low input subplots just received a pre-plant application. So we found, after sampling our weeds in each of our subplots, we did this 4 different times with a 0.25 by 0.25 meter quadrat. We found that indeed red clover is effective at suppressing weeds. So here on the x-axis, we have our subplot types. I'll remind you that PC stands for plasticulture, RC stands for red clover, and we have our chemical input subplots denoted with H and L. So that's a high and low. So you can see the red clover is the weed counts in red clover plots. Both of them are significantly reduced compared to our average weed count of the plasticulture plots. And I'll just say that broadleaf is represented here in red, grass is represented here in green. And here you can see kind of a real world example of that. These were our plots over the summer. Far left is the PCL plots, mainly Johnson grass grew here, as well as morning glory. And then on the far right, we have our red clover plots, which the dominant plant here was definitely red clover. So I mentioned the dominant plant here was red clover and our red clover plots. So they may have been, the red clover may have been a little too competitive. And this was reflected in the fruit yield that we observed. So same deal on the x-axis with our PC and RC, H and L subplots denoted with these monikers. The RC plots had a significantly lower total weight in kilograms for the summer. Total weight represented in blue, our marketable weight represented in green, our cull represented in red. So yeah, so we're thinking that maybe the red clover is outcompeting cantaloupe pretty significantly. So what are we going to do about that? That's really not good for the farmer. The whole point is to grow as much food as possible for your land. Because we think that the red clover is outcompeting the cantaloupe, we're considering making changes to our setup. So these would include making the crop rows wider to minimize the clover competition. This previous season, we did 24-inch spacing for our crop rows, but this coming summer, we plan on doing 40-inch spacing. We plan to strip till deeper to really get all the clover out of the crop rows. So this previous season, we simply used a hand tiller to establish our strip rows. But this coming season, we'll probably be using a real rotavator. And we may decide to mow the clover closer to the planting date to additionally reduce competition. So with that, I'll end my talk. Thank you so much for listening. Again, my name is Leo Kerner. You can contact me through these emails.
Great. Wonderful. And so we have until the top of the hour, so 8 minutes, and we have some questions that have come in on the chat. So for Jaime and Kurt, and so I will go for a couple of questions with Jaime first, and we'll see how we do on time. So Jaime, if you want, there we go.
Yes.
Will multi-grafted trees become a source of pest infestations?
Okay, I am reading a second one. Okay.
Oh, well, we could go with the second one. Okay. So the second one would be the grafted trees will be attracted, and then the grower will use a trap and kill system, ? So.
Well, thank you. Because I came late, I am going to answer to this one, which is, have you determined how many of those trees are needed to achieve maximum protection? And then I will go into there too. Well, basically, at this moment, I am proposing using those trees as a monitoring sites. We have done work, or we haven't done work to protect the block. So my point is that I would like to be able to use one tree in the perimeter for monitoring. The work that I showed, I know it shows multiple trees in the perimeter, but I am not there yet. So I need to wait maybe 1 or 2 more years before I can tell you to what extent we can protect the crop using multiple trees. So in short, I am suggesting one single tree for monitoring.
Okay.
Thank you.
All right. Okay, great. Great. And I can probably put the questions that maybe you can't see because you just came in
Yeah.
I'll put them into the Q&A for you, and then you could answer them from there. So, all right. So Kurt, if you're available, there's a question for you. Why did you choose paraquat for termination as opposed to using glyphosate? Paraquat would seem to be a much greater risk.
We chose paraquat because we had herbicide resistant or glyphosate resistant weeds present in some of our fields. Another thing we chose to do was using that shielded sprayer, and those applications were actually made before flowering or before the vines were running off the plastic in order to avoid any danger to potential pollinators during the application.
Okay, great. Thank you. And there's a couple of questions for Leo, if you're available. The first is, are there other, oops, my screen keeps moving. Are there other cover crops that you've been considering for row middle based on your experiences with red clover?
Hi, yeah.
Uh-huh.
Yep, I'm here. Sorry, yeah. So for this project this summer, or this past summer, and then this summer to come, my project is really only considering red clover. So we're really just looking at this one species and this one cultivar. Not only to look at weeds, but we're also looking at the insect communities using just this one cultivar. So we're kind of limiting our focus. Yeah.
Okay, great. And there is another question for you from Gary Fish. If you make your strips wider, won't that require more land to produce similar yields?
Yes, that's correct. So we are increasing the row spacing, or I guess we're increasing the width of our crop rows to 40 inches. And so with that, we're also increasing the total area of each plot as well. So I think we did a 45-foot width this past summer, and I think this coming summer we're going to be increasing it to 50 feet width.
Okay, great. And for Long Hee, we have a question for you. Do you have data or information on the cost of your system?
So yes, we have a kind of estimated like cost for the systems. For the first one, that's the Air Plus sprayer one, that the intelligent kit is normally cost a bit 30 to 40,000, and then put in your sprayer. For the robots that I showed there, so the one that the second one is I mean, the both second, third one is pretty much like $20,000-$30,000 for the base robots, and then that's, I think that's pretty much the cost, and it depends on the size. Sometimes if you have big field, you want big size the bigger size sprayer maybe cost a bit more. So that's pretty much like $20,000-$30,000 for that type of smaller robotic sprayers.
All right, wonderful. I think we're going to move on because we're almost at the top of the hour. And if folks can take advantage of the Q&A system, it makes it a lot easier for me to track the questions than it does in the chat. And I will try and move the questions that were put in the chat that weren't answered into the Q&A. So, all right. So our next presentation is for Megan Shire, and it's operationalizing eDNA technology for disease vector mosquito surveillance and control. It was funded by the Northeastern IPM Center, and Megan is with the University of Maine.
Hi, everyone. Thanks for tuning into my talk today. My name is Meg Shire. I am presenting on behalf of our PIs, Dr. Ali Gardner, Dr. Michael Kinnison, and Dr. Karen James, on our Northeastern IPM Center funded project, operationalizing eDNA technology for disease vector mosquito surveillance and control. And this research was conducted out of the University of Maine, conducted in collaboration with the Maine eDNA group. And so what I'm going to tell you about today is the experimental design of our study. But first, I just need to get some definitions out of the way. So I'm going to be referring to eDNA throughout the project, which is environmental DNA, and this is genetic material left behind by organisms in the environment. This genetic material can be detected by taking environmental samples, filtering substrate from those environmental samples, extracting DNA, and performing some qPCR or metabar coding. This has been done in samples like snow samples, water samples, soil samples, and even air samples. And there are many applications for this techniques. We can ask questions like, is this here, about a specific organism or taxonomic group? We can also ask questions like, what is here, using bioinformatics or metabarcoding. But in our project, we want to use eDNA for mosquito surveillance. And the reason that we think eDNA can be useful for mosquito surveillance is because we really rely on effective mosquito surveillance. It's how we measure control efforts when we implement larval or adult mosquito control. We measure the effectiveness of that control through mosquito surveillance. We also don't have vaccines for for many mosquito-borne diseases, meaning that there's a high public health reliance on vector mosquito species surveillance. And even though we rely really heavily on it, there's a few major problems with the surveillance. It takes a lot of time and personnel. You need the specific taxonomic expertise. And it's not always useful for cryptic species. Some mosquito species can breed in small aquatic habitats that are really tightly associated with residential environments, some of which can reproduce in aquatic habitats smaller than a teaspoon. And there's a lot of variation in mosquito management across states and counties. For example, in Maine, there is no state or county level mosquito control. It's all up to individual towns. And lastly, vector mosquito species suitable habitat ranges are shifting. This means there's the potential for mosquito-borne disease emergence in new areas. And a lot of our disease vector species, including Culex pipiens and Culex restuans, which are the principal vector of West Nile virus in the eastern United States, along with Aedes albopictus and Aedes aegypti, which aren't up in Maine yet, but are very prominent disease vector species in other places in the US and around the globe, are container inhabiting species in their larval stages. That means the adult females are laying their eggs in these small aquatic habitats, and the eggs, larvae, and pupae are all developing in those aquatic habitats. And so while this presents a challenge for mosquito surveillance, we also think it presents an opportunity for aquatic eDNA sampling. And the purpose of this is hopefully for the adoption of these methods by mosquito abatement companies or groups, whether those are companies or state organizations. And so our research objectives were really in three-part, but what I'm here to tell you about is how we optimized a hierarchical occupancy model design in order to optimize our probability of mosquito detection in container sampling. And the way we did this was we set up an experiment with all of these different levels. And so in this experiment, we wanted to know the probability of detecting mosquitoes at the site level. On each of these sites, we deployed 2 buckets and allowed mosquitoes to colonize those buckets. And when it came time to take our samples, we took 3 samples from each bucket. And when it came time to perform qPCR on those samples, we did that in 4 qPCR reps. So essentially, what we were doing here is taking into account the probability of making a type 2 error that is in this system a false negative or a non-detection of mosquito DNA from a positive sample. And from each level, in calculating this probability of making a type 2 error, ultimately, we are able to calculate this variable psi, which is the overall probability of detection, giving non-detection probability at each prior level. And it's really this rigorous quantification of these methods that we think makes this is going to make these results useful to mosquito abatement and other control groups. Our current directions right now are working on a mitochondrial assay as opposed to a nuclear assay. And I'm really excited about this work because I think it has applications for people and policy. Here's my contact information if anyone is interested. Thanks again for listening.
Wonderful. Thank you very much. So I've determined, I think there are issues with the Q&A and the chat this morning. So if you can use the chat, use the chat. If you can use the Q&A, use the Q&A. I think we're just going to have to go with the flow on it. So Dwayne Joseph is up next. Between and within, utilizing biosolarization and living mulches to manage weeds and vegetable systems. The research was funded by ARDP and Dwayne.
This presentation will go over some early findings from one of my current projects evaluating the use of biosolarization, strip tillage, and living mulches for weed management in vegetables. An integrated weed management program involves using a combination of tools to establish a comprehensive approach to managing weeds. Currently, there is a need for integrated weed management programs, and that's mainly because of an increase in herbicide resistance in weeds. If we look at vegetable systems, there are two main areas where weeds need to be managed if we want to protect yield: within the row and between the row. These two areas are treated differently for obvious reasons, and both present different challenges related to weed management. In this study, we evaluated 4 treatments consisting of different management practices for within and between row weed management. In our study, we used cover crops in 2 main ways for weed management. Red clover was grown between the rows of all our treatments as a living mulch, except in our solarized pseudo control treatment. And this was done mainly because of clover's ability to suppress weeds via competition. And it can also be used to suppress insect pests and attract pollinators. Within the crop row, cereal rye was used as a terminated or die-in mulch. And this is because cereal rye produces a great biomass that forms a thick mulch on the soil surface when roller crimped in the spring. The cash crop can then be directly transplanted into this mulch as it suppresses weed germination and emergence. Another method for within-row weed management evaluated in the study included using biosolarization. And this is a soil disinfection technique that is similar to solarization but involves using an organic soil amendment prior to solar heating. As the moist soil heats up, there is a release of phytochemicals that have been shown to inactivate weed seeds in the soil. The addition of the organic soil amendment allows biosolarization to be effective in a fraction of the time that it takes solarization, and in regions less conducive to solarization. In this study, we used a mixture of apple and grape pomace, which are the byproducts or waste generated from fruit processing. Pomace is a promising biosolarization soil amendment because they are rich in organic compounds, they don't pose any biohazard risks like animal waste, and are relatively abundant and inexpensive. So, does it actually work? Yes. So if we look at this graph that shows between and within row weed abundance in response to treatment, we see that within the row there are less broadleaf weeds and grasses in biosolarized plots early in the season compared to the other treatments. However, does it hold up throughout the season? Not really. While there are no broadleaf weeds in the plot, there was a substantial number of grasses and sedges late in the season. So why did we observe this? If we look at the structure of nut sedge, and in this example yellow nut sedge, we'll observe why biosolarization wasn't very effective on them. And that is mainly because they reproduce and spread vegetatively through tubers on nutlets and not by seed. And if we remember from earlier, it has been documented that the main mode of action for biosolarization is weed seed inactivation. Also, the increased soil disturbance in preparation of the biosolarized plots may have spread and contributed to the high density of nut sedge that we found in our plots. So in summary, biosolarization is a promising weed management technique. We saw how it effectively managed early season weeds, especially broadleaf weeds. Also, it can be used with other techniques as part of an integrated weed management program. It can be used also as an organic pre-herbicide alternative because it can provide early season weed control before canopy closure, allowing the crop to gain a competitive advantage over the weeds. Currently, more research needs to be done to evaluate the biosolarization technique, along with the efficacy of different sources of pomace. But what we can say is that biosolarization is not effective on nut sedges, and that is mainly because of its vegetative reproductive habit. So with that, I'd like to acknowledge our funding source and all the other collaborators on this project and the PI on this project, Cerruti Hooks. My name is Dwayne Joseph, and this is my contact information. Thank you.
Wonderful. Thank you very much. So we have a question that has come in for Megan, and give people time to pop up a question for Dwayne. Do I understand, this is from Deb, do I understand that you obtained your samples by allowing for natural populations, and natural is emphasized, in your sample buckets rather than inoculating water with different species?
Yeah, thanks for the question. So you did understand that correctly. For the for the experiment that I shared, we allowed those buckets to be naturally colonized by the mosquitoes we were looking at, specifically Culex pipiens. This was informed based on some research I had done the summer prior. So all of those 5 sites where we deployed buckets were sites that we had collected Culex pipiens in the year past. We did do another experiment where we took samples from closed containers with known numbers of Culex larvae to try to understand if there's some threshold of detection. So we are looking into, we explored experiments with natural container colonization, as well as with some known numbers of mosquito larvae too. So happy to talk more about this offline as well.
Okay, great, lovely, wonderful, thank you. And we do have a question that came in for Leo that we didn't get to earlier, so maybe we can pop back to that if Leo's still available. The question is, did the cover crops cause problems for the main crop in the dry years?
Yeah, thanks for the question. Yeah, so this year actually it was relatively dry, especially between March and June. So yeah, it still was causing problems despite the dry season, and I think this is because so we seeded the clover in October, and so it kind of overwinters, and so maybe builds up some resources during that time, and then based on previous research, there's not that much, I don't think, on red clover itself, but other clovers, like sub clover and white clover in particular, I think were 2 that I've read. Those seem to be less competitive, and I think especially in the dry season as well. So, yeah, thanks.
Great, wonderful, and we will move ahead. And the next presentation, Fry, and his title is "IPM is for Everyone: The Reach and Impact of a Virtual IPM Education Series." It was funded by the Northeastern IPM Partnership Program, and Matt is with the New York State IPM program at Cornell University.
My name is Dr. Matt Fry, and I'm an extension educator with the New York State IPM program at Cornell University. Today, I'll be giving an update on a 1-year IPM communications project entitled IPM is for Everyone: Enhancing the Reach and Impact of a Virtual IPM Education Series. This project was undertaken by Dr. Amara Dunn-Silver, Dr. Alejandro Calixto, and others from the New York State IPM program. Before I talk about the project, I wanted to introduce our virtual series that began 1 year prior to the awarded funds. In March of 2021, we launched What's Bugging You First Friday to increase public awareness of IPM and reach audiences isolating at home during the COVID-19 pandemic. The series offers timely topics in short, engaging presentations that focus on actionable items. 30-minute sessions are divided into a main presentation and an IPM Minute, which highlights actions to take for the weekend ahead. Sessions take place on the first Friday of each month at noon, and recorded presentations are posted to YouTube for asynchronous viewing of content. In our first year, we promoted events through familiar channels within our networks at Cornell University and Cornell Cooperative Extension. We were thrilled to see over 650 people register for events, but noticed that attendees were clumped in the Finger Lakes and Hudson Valley regions of New York, and they predominantly identified as white or Caucasian. These findings highlighted a need to expand our audience, considering that topics apply to people wherever they live, work, learn, and play. Our IPM communications proposal was funded for March 2022 to February 2023 and included several objectives. First, we wanted to expand and diversify our audience and presenters. We relied on social media to reach new audiences by promoting events on Facebook in the Northeast. In addition, we took a more active approach by joining community and interest groups on Facebook to share event details directly with those pages. We also asked state IPM coordinators to distribute information about the series. To identify speakers outside our circles, we searched faculty pages of institutions throughout the Northeast. Unfortunately, this was a time-intensive and difficult process because staff pages do not always provide details about expertise, or if staff are available for extension presentations. An important objective of our proposal was to increase efforts to reach Spanish-speaking audiences. To this end, we offered 1 session in Spanish in 2022 and 4 sessions in 2023. We created a new landing page for Spanish programming and a dedicated YouTube channel. An objective we were really excited about, both for the potential to entice event participation and also to document adoption of IPM principles, was a photo contest. The premise was that attendees would submit a photo of themselves performing an IPM task, and we'd send a prize to the selected monthly winner. The funding allowed us to create really nifty prize packs, and we promoted the contest on social media and during live sessions. Unfortunately, contrary to our expectation, participation in this contest was low, and we had to directly solicit photos to ensure we'd have at least one to share each month. Our final objective was to share insights about developing an accessible virtual program, which we did through the development of a 3-page fact sheet available on the New York State IPM program's website. The document is organized chronologically from planning the series to action items needed before events, during, and after. This funding helped us to increase the geographic footprint of our programming. Specifically, we saw nearly a 110% increase in the number of zip codes represented by attendees, and the number of participants continues to grow. Importantly, attendees are learning new things that they have implemented or plan to implement, and most attendees indicate that they would share First Friday series with their friends or colleagues. In terms of our next steps, we hope to further expand our audience so that attendees better reflect demographics. For example, we currently have low registration from major cities in the Northeast. As mentioned, we also hope to better reach Spanish-speaking audiences. While the number of people registering for events, attending live sessions, or watching recordings continue to grow for the English sessions, registration and attendance at Spanish language events remain low. We hope to improve both aspects through collaboration in a subsequent proposal. Thank you for your time.
Wonderful. Thank you very much. And our next presentation is by Emma Weltman, and she is with Rutgers University. And the title of her presentation is Exposure to Insecticide Drift in an Insectary Strip Does Not Negatively Affect Survival of Trisoculus japonicus. And this was funded by the Northeast SARE.
Hi everyone, my name is Emma Waltman. I'm a PhD student at Rutgers University. My talk is Exposure to Insecticide Drift in an Insectary Strip Does Not Negatively Affect Survival of Trisoculus japonicus. Insectary strips are unsprayed strips of flowering plants that attract and retain beneficial insects. Here I have a diagram of a buckwheat insectary strip, which is the flowering plant that we looked at in our city, surrounded by peach orchard blocks. These strips offer nectar and pollen sources to beneficial insects and also refugia from agricultural management in surrounding habitat, and this is why they attract and retain beneficial insects. The strips are also beneficial to farmers because they become a source of beneficials and then can help you mitigate pest pressure. The management of surrounding crops to the insectary strip, however, could expose insects in the insectary strip to insecticide drift. And this is obviously going to reduce the effectiveness of the insectary strips at giving you good populations of beneficial insects on your farm. We specifically wanted to study Trisoculus japonicus and look at the effect of insecticide drift on the survival of that parasitoid. We chose it because it's a really relevant parasitoid right now. I'm sure many of you have heard of it. It's a main candidate for biological control of Polymorphohalis, or the brown marmot stink bug, in North America, where we have inventive populations of it. And field studies have reported variable control, but up to 25% parasitism of sentinel eggs. The study objective was to determine if the insecticide drift affects survival of Trisoculus japonicus. To do this, we seeded buckwheat strips adjacent to peach orchard blocks at our research center in Bridgeton, New Jersey. We applied Actara or thiamethoxam with an air blast sprayer at 4 different times in 2021. And we had 3 treatments of buckwheat to control buckwheat, drift buckwheat, where half of the sprayers were turned on only to directly spray the peaches, and spray buckwheat, where both the peaches and the flowers were sprayed. After spraying, we cut buckwheat flower sprigs and also peach tree leaves from unsprayed and sprayed trees to set up a survival assay for Trisulcus japonicus. We introduced Trisulcus japonicus in groups of 10 to buckwheat and leaves and monitored their survival in the lab. And we did this in plastic tubes. So we have these 3 treatments: the control buckwheat, drift buckwheat, and spray buckwheat. An additional 3 treatments with a control buckwheat, control leaf treatment, control buckwheat, spray leaf treatment to kind of simulate what actually would be encountered by Trisulcus in the field, a spray buckwheat, spray leaf treatment, and then just a sprayed leaf treatment without any buckwheat. And here are the results. Here's the probability of survival to 24 hours with 95% confidence intervals as generated by a binomial generalized linear mixed model. To break this figure down a bit, you can see that Trisulcus japonicus survived equally well on control buckwheat and drift buckwheat, and also control buckwheat, control leaf. This is great. This tells us that the amount of insecticide on the drift buckwheat treatment was not enough to cause mortality. Additionally, you can see that the Trisulcus japonicus wasps had better survival to 24 hours when they had a buckwheat food source, even when they were exposed to direct insecticide, as they had higher survival than just on the spray leaf. In conclusion, insectary plants do fit into existing management practices. The thiamethoxam was not enough to kill Trisulcus japonicus in the drift treatment, and the buckwheat food source prolonged survival of our wasps, even when exposed to a direct spray. I want to thank my funding sources and Rutgers for applying insecticide sprays, the summer 2021 lab team, Max McCarthy, for analytical support, and I look forward to your questions during the question session. Thank you all for listening.
Wonderful. Thank you very much. While folks are gathering questions, and feel free to put them in the Q&A or in the chat for Matt or Emma or Megan or Dwayne or anyone who's spoken I actually have a question for Matt. Is you mentioned having the 2-pager put together about sort of best practices that you found from virtual events, and that it was on your website. I'm wondering if you found some effective avenues for sharing that with other people who do IPM virtual events, other than just putting it on your website, or if you've thought about that.
Hi, Yana. Thank you for your question. Yes, we haven't, really looked at different avenues to share that document. We asked Jerry if she could send it out in the weekly email, and we feel like that's given it some reach, but we're certainly open to opportunities to share that document more widely, and to maybe partner with people that are doing or launching this type of programming. If they have any questions, we've provided our contact information there as well. So we're interested in expanding the reach of that document to help people create better and more accessible presentations.
Yeah, I'm actually thinking it might be good to share it with the regional centers across the US and asking them to bring it, because it's applicable all across the US and might be a good case. So, all right. There's a link actually in the chat to the documents, it looks like. We also have a question for Emma, if Emma is available. And the question is, would you please explain the spray leaf purpose again?
Sure. And I sent a response, but I realized I only sent it to the panelists, so probably not everyone can see it. But basically, I'll just kind of read what I wrote. So it's likely that the parasitoids that would occur on a farm would probably come in contact with directly sprayed peach trees or any sort of crop that's being directly sprayed with insecticide. So basically, we wanted to see if having a high quality nectar source in the form of the buckwheat flowers could prolong their survival even when in contact with direct spray. So that was the point of looking at like their survival with the buckwheat with the directly sprayed leaf compared to when they just had the directly sprayed leaf and no nectar source. Hopefully that answers your question.
I think so. I think it does. Thank you. And all right. And I am not seeing any other questions right now. So I'm thinking that maybe we should move ahead. Please feel free to put your other questions in the chat or a Q&A. So next we have Umara Dunn-Silver, and the title of her talk is Increasing Awareness of Biocontrol as a Part of IPM through Digital Outreach, an Update on Progress. This was also funded by the Northeast IPM Center, and Umara is also at the New York State IPM program at Cornell University.
Hello, my name is Umara Dunn-Silver. I'm the biocontrol specialist with the New York State IPM program. And today I want to give you an update on an ongoing project to enhance some digital resources related to using biological control. So this is a project that is migrating information from this biological control website that was originally created by Tony Shelton to the New York State IPM website. After Tony's retirement, arrangements were made for us to take over management and updating this information. So my goals here are to, first of all, improve the usefulness and usability while we're doing this migration, and second, to create and add new content. I've broken this work up into three phases. Phase one is complete, and that is focused on what is biocontrol, some of the basics, kind of surface level biology, and then a real emphasis on practical information. How do I use biological control and resources that help people use it? Phase two is just beginning, and this is migrating articles about individual biocontrol agents. So there are many, many articles about individual biocontrol agents written by authors from across North America. And I've been getting a lot of support from some of those past authors and some new authors in reviewing and updating those articles and getting them ready to put on the new site. And then phase three will be everything else. This is a quick overview of what the new site looks like. So you'll see this big picture of a lady beetle when you first go to the page. This QR code in the upper right corner of the screen will take you to the site. And I also have the URL down here. And I'm going to take just a minute to explore this with you. So here we are at the page. I want to focus on these three main areas first. So as I said, I've put a lot of emphasis on the practical, how do I use biocontrol? There's some more information on the biology behind biocontrol, and then this link will take you to the beginnings of those biocontrol agent profiles. So let's start here. There's some new resources here that I want to make sure to mention. So some specific resources I wanted to highlight. If you have not already seen our pocket guide for recognizing beneficial insects, especially natural enemies, it's a great resource. It's printable, lots of pictures, both in English and in Spanish. I've also been putting together a lot of resources on helping people use biopesticides more effectively. So there are summaries of university research looking at efficacy of these products for different crop groups, except I haven't done vegetables yet. And then these biopesticide profiles are focused on the biopesticide projects themselves. They're just 2-page printable PDFs that look at some of the practical details like storage conditions, shelf life, mode of action, so that if someone is using these products, they're making choices that will make it most likely their project will succeed. There's also a big section on conservation biocontrol with a focus on creating habitat for beneficial insects. This link will take you to really detailed step-by-step instructions. There's also some shorter and some longer lists of plants that might be relevant here. And then the last thing I wanted to show you was a quick introduction to some of the biocontrol agent profiles that are available. One of the first ones we did was for this sulfur knapweed moth. And I've tried with these profiles to break the information up into smaller pieces, make it easier for people to access, put the information that people are most interested in knowing first near the top, things like, does it work? Where should I use it? Again, you can see a shift to bulleted lists as much as possible in shorter paragraphs, pictures, but we've still retained all of those great details that were in the original articles down at the bottom of the page in this Learn More section. And that is my quick overview of what's going on with this website migration. All my contact information is here, as well as the URL for the website. And a big thank you to all of the authors who are helping me review the content for these individual biocontrol agent profiles. Thanks so much.
Great, that was wonderful. It's a beautifully laid out site. So our next presentation is Lynn Sosnowski, and her title is Integrated Weed Management in Hemp, a multi-state effort to evaluate practices and develop recommendations. It was funded by ARDP, and Lynn is at Cornell University.
Thank you for attending this talk, Integrated Weed Management in Hemp, a multi-state effort to evaluate practices and develop recommendations. This is a CPPM grant that was funded in 2021. I'd like to recognize our National Research and Extension team. I'm Dr. Lynn Sosnowski at Cornell University. My co-PIs are Dr. Matthew Catul at Clemson University, Dr. Harleen Hatterman-Valenti at North Dakota State University, Dr. Michael Flessner at Virginia Tech, and Dr. Carla Gage at Southern Illinois University. Why hemp? The demand for hemp-derived products, including textiles and fibers, food and feed, personal hygiene, and nutraceutical and pharmaceutical products is growing. However, there is a knowledge gap for hemp production. The prohibition against growing cannabis also banned research on best agronomic practices in hemp, including the development of weed management recommendations. Consequently, hemp growers are producing hemp with a fraction of the information usually provided to other commodities. More than 70% of respondents of a USDA NIFA supplemental and alternative crops grant indicated that weed management is a very important to an extremely important topic. The goal of this project is to increase grower knowledge about dual purpose, that would be grain and fiber, and floral hemp cultivar interactions with weeds, and facilitate the adoption of effective weed control strategies. We have 3 research objectives, one in dual purpose hemp and floral hemp. Both of these would look at hemp competitiveness with weeds and define the critical weed-free period as influenced by cultivar and phenotype. For both of these types of hemp, we will evaluate the efficacy of cultural and physical weed management practices. We have a 3rd research objective to look at phenotyping hemp to quantify the crop architectural traits and relate these data to the outcomes observed in objectives 1 and 2. We are also engaged in significant extension outreach efforts to showcase local and regional similarities and differences in our data. This is our progress to date. With respect to the first 2 research goals, we have 4 site years of data in grain and fiber hemp and 6 site years of data in transplanted floral hemp to describe crop weed competitive interactions and to evaluate the efficacy and safety of weed control practices such as altering planting date, altering seeding rate, altering seeding arrangement, the use of cultivation, the use of mulching, etc. in the hemp crop. We have also completed a phenotyping project. This data is being prepared for submission to a journal, implementing digital multispectral 3D scanning technology for the rapid assessment of hemp weed competitive traits. This was done by Dr. Matthew Catul at Clemson University along with his graduate student. Based on the data that we have generated during the past 2 years of the study, we are also engaging in expanded collaborations, one with Dr. Larry Smart's lab to look at seed size impacts on early season hemp and weed competitive interactions. We're also working with the USDA ARS hemp germplasm collection to look at morphological variation in grain and fiber, floral and feral hemp accessions on hemp and weed competitive interactions. With respect to our engagement in the last year alone, we've been very involved in the production of a manual for hemp growers, YouTube videos, lecturing in hemp production courses, and speaking at grower field days. We have also presented our data at a number of professional conferences. One of those presentations was by an undergraduate student working with Dr. Carla Gage, who won second place in the undergraduate poster competition. Dr. Gage also spoke internationally in Morocco on our research. We would like to thank you for attending. We would like to thank USDA NIFA for the sponsorship, and we would like to thank all of our partners associated with this project.
Wonderful. Thank you very much. And I don't see any questions. I do see that Amara put a clickable link for her new biocontrol page if people are interested in that. And so I'm going to suggest that we move along, and feel free to add your questions. It looks like we'll have a little bit of time at the end for that. So the next presentation is by Devamalya Chatterjee, and the title is Novel Maize Germplasm for Sustainable Pest Management. It was funded by Northeast SARE, and Devamalya is with the Penn State University.
Hi, my name is Devamalya, and I'm a postdoc in the Department of Plant Science at the Pennsylvania State University. I'll be talking about our research that focuses on developing and using high flavonoid maize germplasm to control Lepidopteran insect pests. And this research is funded by USDA NIFA, as well as a part of the project was funded by a graduate student research grant that I received during my PhD from Northeast SARE. We all are familiar with the flavonoids. These are the orange, red, and purplish pigment found in different plants. Now, flavonoids are characterized by these 3 defined carbon rings. As you can see in the B ring, there is a position 3' where there is a group R1. Whether the R1 is a hydrogen or it's a hydroxyl group, that can determine what type of flavonoid is being produced. We are highly interested in a type of flavonoid called 3-deoxyanthocyanidin, as well as flavonoids. Now, flavonoids are produced in both maize as well as in sorghum. And the flavonoid biosynthesis pathway in maize is regulated by a gene called P1 or pericarp color 1. And in sorghum, it is regulated by Y1. And in this particular pathway, the product is flavin-4-ol, and at the end, that produces flavins. Now, in case of sorghum, this can also produce 3-DS, and in particularly in sorghum, there is ample amount of flavonoid production in the leaf as well as in the leaf sheath. So we develop a very simple protocol that utilizes acid hydrolyzation as well as organic extraction using isomyl alcohol. The end product that we get is a concentrated extract of flavonoids. Now, we wanted to utilize this use this flavonoid extract against a particular Lepidopteran insect pest called fall armyworm. Fall armyworm became invasive pest in sub-Saharan Africa and have been also been reported in Southeast Asia as well as in Australia. Now, in US, the damage caused by the fall armyworm is particularly during the early stages when the plants are still small seedlings. We are very much interested in our research using the neonates, which are newly hatched larvae, as well as the third instar larvae. So what we did, we used that flavonoid extract that we got from the sorghum biomass, and we supplemented artificial diet. We used a diluted version of this flavonoid extract on detached leaf where we cut the small pieces of leaf from the field-grown plant and spray that with a diluted version of this flavonoid, as well as we spray a diluted version of this extract on the whole plants. And when we feed these three types of diet to the fall army of larvae, what we see, as you can see here in the magenta bar, there is significant reduction in their body weight as well as high increase in their mortality. Now, there is a challenge in case of maize, while particularly looking at these types of flavonoids, because P1 is only expressed in the pericarp, which is the outer layer of the seed. In maize, these flavonoids are not being produced in the leaf sheath or in leaf. So we actually leveraged a spontaneous mutation or natural mutation that's named unstable factor for orange one, which actually overexpresses P1, causing high flavonoid accumulation in the plant. Now, the progeny of this mutant was unstable, but we created near isogenic lines from the plant that produces high amount of pigment, and we call them EV. Now, these EV plants are very interesting because they also produce flavonoid now because of the mutation in the husk, leaf sheath, as well as in leaf. We also created another germplasm where we created transgenic maize lines using the sorghum Y1 gene, because the sorghum Y1 can actually produce this flavonoid in the leaf as well as in the leaf sheath. So now we ended up with two sets of genotype. In one set, EU is the high flavonoid producing line, and in another set, there is the ZMY1, which is a transgenic high flavonoid producing maize line. Now, when we fed the larvae with leaves from ZMY1 and EV, as shown here in the magenta bar and the dark gray bar, you can see there was, again, significant reduction of their body weight and increase in the mortality. Now, the question was, what is actually causing this increased mortality? So to understand that, we dissected the gut. Now, the gut of these insects can be imagined as a two-layer system, more like a trash bin, where the bin is the midgut epithelium and the trash bag is the peritrophic membrane, which is the inside layer actually holding the food. Now, under the scanning electron microscope, we found that when the larvae were fed with both UV and ZMY1 leaf, there was significant damage in their peritrophic membrane. But this damage can be associated with changes in gene expression, changes in microbiome population in the midgut, or changes in the proteomic structure of the midgut. So we are continuing our research further, and I'll acknowledge everyone who supported this research and our grant agencies.
Great, thank you very much. And our last presentation is by Rachel White, and the title is Integrated Pest Management for Control of Gastropod Vectors on Pastures. It was funded by Northeast SARE, and Rachel is with the University of Maine.
Hi, I'm Rachel White. I'm a PhD candidate and assistant extension professor at the University of Maine. Today, I'm going to briefly talk about my research on the integrated pest management for the control of gastropod vectors on pastures. Just a bit of background on snails and slugs. They can serve as intermediate hosts for a couple of problematic parasites, such as meningeal worm, rat lungworm, and liver flukes. I study the meningeal worm system, where that parasite causes problems for livestock producers, as there is no diagnostic test for it. Sheep, llamas, goats, they're a dead-end host for this parasite. So trying to find prevention avenues for these farmers was part of my objectives. So I wanted to have practical strategies that producers can use to reduce the risk of gastropod-borne parasites. I looked at two methods that have been shown to reduce either gastropods in general or other invertebrates. So those are mowing and pastured poultry. For my mowing methods, I used a random complete block design over 2 years. Within the first year, I only had 2 treatments, which was mow and no mow. In the second year, some of the mow sites were allowed to regrow, and that created 3 treatments. Within each plot, there were 6 surveys looking for gastropod abundance, and I also collected environmental variables such as soil moisture and vegetation type and height. And then I used a Kruskal-Wallis non-parametric test, and then in year 2, I did a repeated measures Wilcox pairwise test. For the pastured poultry methods, that was a before and after gastropod count treatment. So there were 3 10-meter lines that I surveyed for 10 minutes each for 8 rotations over just one grazing season. There were about 180 laying hens, and they were rotated about every 4 days. The schematic on the right shows the sampling plots indicated by stars. So the results of both treatments were really positive. So poultry proved to be an effective treatment in reducing gastropod populations with a p-value of less than 0.001. And then mowing was another control strategy that really worked. I did look at the regrowth plots, and those did rebound in gastropod populations almost as much as the control plots. And I did analyze seasonal trends. So over year 2, that's just the image on the upper left there, that did show to have seasonal effect in the gastropod populations. So for my conclusions, pasture poultry exposure across gastropod abundant pastures can lower snail and slug populations. I encourage producers to use active pasture hardy breeds. And another recommendation is to perhaps use poultry before small ruminants and other livestock get moved onto pasture. And that's sort of backwards to other IPM strategies where you'd introduce poultry to scratch up manure to really like spread it within the field and to eat the fly larva that's on the other livestock manure. So that recommendation is a bit backwards. And then mow or short graze for at least 2 years, or just have a combined effort of pastured poultry and mowing. Thank you very much. I know it was very brief. If you'd like to learn more, my email is up at the top there. Thank you.
Great. Actually, I think that email was cut off by the logo, but I'm sure we can make it available to you. So that is the all the live presentations we have. I can see there's a conversation going on between Dwayne and Lynn about hemp, and so I don't see any open questions. So what I'm going to do is move along, and I just want to remind people that we have a Find a Colleague site, which I think is very helpful for people, and that is where you can post a profile of yourself and the kinds of things you're interested in, and you can also go to this website and see colleagues who have already posted about themselves. It's a great way of interacting with people that across the Northeast. And I looked at it the other day, and over the years, it really has populated. So if there's folks here that you're interested in their work, go see if they have already put a profile in, or put one of yourself and use it to reach out to them. There is a question here for Rachel. Did you look at the density of parasites versus the density of poultry needed for control?
Hi, great question. So if you're talking about parasites in general, in this system, or both these treatment systems, I didn't actually collect the gastropods and digest them to quantify the parasite numbers within them. I did do that, however, on these farms in totally separate fields, just to actually assess the parasite numbers that were on these farms. So that was a totally different chapter. So I guess in short, an answer to your question is no, but that is something I can look into. And I think that is an interesting question. So if I compare it against the parasite numbers that I found on these farms, but where these poultry were not housed.
Great, lovely. Thank you very, very much. And I'll keep my eye out if there are any more questions that come in. We do have a poll for you with some questions about how helpful this session has been for you. So it should pop up on your screen. There it is. And so I'll just be quiet for a moment. There's just 4 questions. They're pretty simple. All right. I think we have a preponderance of people that have answered the poll. And I assume some other people probably are driving or not available to do that. So I will end the poll and thank you for doing that for us. And there will be an archive of today's webinar, and you'll be able to watch the recording as often as you like. And I will send you an email with that. Sorry, I'm just keeping an eye on the questions here at the same time. And that concludes our conference for today. But we have another slate of people who are speaking on Thursday, same time, 10:30 until noon on Thursday, same link. And so if you enjoyed today and found it helpful, please feel free to join us on Thursday. And thanks for everyone's participation. And you just got an extra 8 minutes in your life. So, well, okay, we'll see you Thursday. Thank you.